This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1057, recorded on October 27th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, Vincent. We've got 86 degrees and partly cloudy. We've had a lot of rain recently, which is welcome, but it doesn't go it doesn't make up the deficit much, which is substantial. We are entering winter. As a matter of fact, next week they're looking at one night with an overnight low of 32 degrees and a high in the mid 50s. So summer's over.
Here in New York, it's 23 and mostly cloudy. Also joining us today from Madison, New Jersey, Brianne Barker.
Hi, guys. Great to see you all. They tell me it's fall and getting close to winter, but the weather here doesn't seem to be aware of that. It's 74 Fahrenheit. And while my phone says partly cloudy, I see an awful lot of sun at the moment. So it does not seem like the end of October here at all, but I suppose I'll take it for a couple more days.
So we're at the Incubator today, of course, and I have a guest for you. He is from Oregon Health Sciences University, Klaus Früh. Welcome to TWiV.
Very well said. Thank you. Thank you for having me. It's a real pleasure.
Yeah, I practice Früh a lot because I like to get last names correctly. Klaus, you're visiting New York, right?
Yeah, that's right. Gave a talk at NYU yesterday. I have two friends there. One is Dan Littman, who I go skiing with once a year in Jackson Hole. And the other one's Ian Moore, who I play music with for our band, Herpetic Legions, that performs at the International Herpes Virus.
Oh, I've seen Herpetic.
Yeah, so Ian's our drummer. I'm one of the many guitar players.
Because I saw them at ASV, but you weren't there. It was.
I was at ASV, but when
this last one, but many years ago.
Oh I was.
It was
there,
Ian. And it was, I think Rick Lloyd sat in.
Yeah.
And then a couple of herpes people from Cornell.
Yeah.
I can't remember.
But I've never been professionally at NYU, so that was nice.
Good. Very good.
So, Klaus, what department were you visiting?
Oh, I don't even know what the name of the department is. Whatever Dan Littman is heading up.
Yeah.
Okay. Do you know Ed Ziff?
No.
Okay.
So, Rich,
old friend of mine from NYU.
Rich, the classic department names, microbiology, biochem, forget it.
Yeah, okay.
It's like molecular medicine, molecular this, it's just whatever thing they can come up with.
I was going to guess immunology stuff.
Immunology stuff, yes. Department of
That's
Immunology
the name of the
stuff.
department, immunology stuff.
I come across so many creative names. Anyway, Klaus and I were on the bus back to the airport after ASV in Athens, and he sat next to me and introduced himself, and I said, well, if you're ever in New York, come on by, and he did. And I was in here setting this up, and he walked in, and then two people walked in behind him, who I assumed were with him, but apparently they were just independent people who decided to visit the Incubator at exactly the same time that Klaus walked in. And one was a nursing student at NYU, and the other a boyfriend from Oregon, which is funny. Right?
Yes.
A medical student from Oregon, yeah. So we have a picture of them here at the Incubator. All right, before we get to talking with you, Klaus, a couple of items of business. First of all, if you like our work here, doing science communication, please consider supporting us. You don't have to give a huge amount of money. You could give $1 or $2 a month, which is less than a cup of coffee. As Angela would say, it's less than a cup of bad coffee, right? You can go to microbe.tv/contribute. We have a number of ways you can help us spread the word for science. I also want to remind everyone, Amy Rosenfeld's lab at the FDA has a second research assistant position available. Just to remind you, her laboratory works on enteroviruses, including enterovirus D68. She's interested in pathogenesis. cross-reacting immune responses, and animal models. And all this work is done under BSL-2 conditions, and so the technician would help do that. You can go to the show notes. We'll have a PDF there, and in that is a description of the job position as well as her email, so you can contact her. And finally, it's the last time we're going to be announcing this. Are you going to any Halloween parties this year? Do you have a costume in mind? Well, you might want to check out the costumes at vaccinated.us because they have spike shirts. And if you buy one of those as part of your costume, part of the profits will go to Microbe TV to help support our work. And as you know, vaccinated.us has helped us many times before. And so we'd love to help have you help us again. And so the idea here, and you have probably seen me and Rich and Cindy Leifer and Jolene Ramsey and even Daniel Griffin dressed up in our Halloween costumes with spike shirts and capes and masks. And thank you guys for doing that. I know it feels a little funny. I went with Daniel in Chicago to his hotel room and he put on his cape and his mask and his spike shirt. But those are all just to show you how much fun you can have putting a costume together.
I haven't seen my superhero picture posted.
They look very fun.
Dude, you haven't seen yours posted?
No.
Oh, Karen, we have to get it posted.
I got an email with one in it, but I haven't seen it posted anywhere. I was even poking around on Vaccinated US. There almost ought to be a little channel there with them, or somewhere on Microbe TV or something. At any rate, I have a chorus rehearsal on Halloween, and I'm going to wear my science superhero.
Oh, that's great. That's great.
Yeah, absolutely.
When is Halloween? Like Sunday or something?
Tuesday.
Oh, I'm going to be... Can I wear it on the plane to Montreal?
Yes. Oh, absolutely.
Yeah? Karen is nodding. Yeah. Oh, I like having an assistant here. It's like Johnny Carson talking to his assistant off stage. It's like, hey, Ed, is that what we can do? You remember Johnny Carson?
I didn't grow up in this country, so you didn't grow up with Johnny Carson, but I know
Oh,
who he
my
is.
gosh. I'm going to wear my spike costume on the plane to Montreal. That'll be fun. And then we'll go out for a drink that night, Karen, and we'll wear them. All right, very good. But the idea here is, the costume is a superhero. And this year, we're all indebted to the superheroes who helped get us through the pandemic, right? All the healthcare workers, all the scientists, vaccine developers, and so forth. They're all superheroes, so we want to acknowledge that. And so please go to vaccinated.us. They'll tell you how to build your costume. One more thing. You have to enter Microbe TV at checkout because that will direct some of the profits to us. And you will, I don't know now if you're going to get your order in time for Halloween unless you expedite it because you might be hearing this on Sunday and Halloween is Tuesday. But nevertheless.
Listen, it's good for next Halloween.
Good for next Halloween. You can always wear spike shirts.
Yeah.
And hey, if somebody in a cape shows up on the streets of New York, nobody would notice. Right? Isn't that true? And we thank you for your support. And we thank Matt at vaccinated.us for doing this because it really helps us to do our programming. All right, Klaus, let's talk a little bit about your history first. So tell us where you're from and where you got educated and all that.
Yeah, so I grew up in a very small village, somewhere between Freiburg and Strasbourg in the very southwest of Germany along the Rhine River. And, when I was a kid, it actually was still a working farm. My grandparents had a farm, and so we had pigs and cows. And one of my oldest memories is the first time I looked into a microscope. Because when you slaughtered a pig, you would actually slaughter it at home. So a butcher would come, you kill the pig, everybody helps. By the end of the day, the pig is converted into schnitzels, right? But every time you did that, the health department would send an inspector to check for trichinella. Mm-hmm. And so the guy would come and, make slices and then put them under the microscope. And I was a little boy, I was like really curious what the guy was doing. I said, here, you want to look into the microscope? And I still remember how it looked like, the staining and everything. So I was always a curious kid. And then, wanted to study biology, did go to university, then ended up at the University of Heidelberg and did my PhD with Hermann Bouchard. So he was an old-time molecular biologist, was trained here in the US, with people like Gobind Khurana, where they were working on deciphering the genetic code. So he was one of the leading molecular biologists, but what I was doing in his lab was quite different from the rest of the lab. The rest of the lab was into gene regulation. You probably all know the TET inducible system that was developed in this lab. It's now used in thousands of labs across the world. I was working on malaria vaccine. So he had developed expression systems, and basically the idea was, can we develop a vaccine against malaria using E. coli expression and so on. But what I did is so he sent me to Africa, to Mali, to do field studies. So taking blood from people and then, studying the immune response to malaria over time. And in Mali, like in all the Sahel area, malaria is very seasonal. You have a very strong wet season, you have a very strong dry season. So during the wet season, mosquitoes, malaria. During the dry season, no mosquitoes, no malaria. And what was interesting is that you could clearly see how nobody was really immune to malaria. Okay, so they had very strong antibody responses, very strong T-cell responses, but they got infected again and again. And you could see how the antibodies went up in the wet season, went down in the dry season, so they got also sort of boosted every year. So my lesson from that was that just because something is immunogenic doesn't mean it's protective.
Oh, how about that?
Yeah. and so
This is how many years ago?
In the 80s, okay, so quite a while ago. So I decided for my postdoc, I wanted to go more into really basic immunology, understanding more how the immune system works. And I joined a lab at the Scripps Research Institute in La Jolla, run by a Swedish person, Per Pedersen. And Per was a biochemist, but was studying antigen presentation. The way he came to antigen presentation is also interesting. So Per's claim to fame was in the early 70s, he sequenced beta-2 microglobulin, which, was just a protein that you could find and isolate from urine. And so they sequenced the actual protein before this was before you could sequence genes and all that, not knowing what it was good for. And of course, now we know it's a subunit of MHC class 1 molecules. And so he became an antigen presentation guy because, he sort of discovered or sequenced beta-2 microglobulin. And when I joined his lab, this was a very exciting time. So beta-2 microglobulin was discovered, histocompatibility molecules were discovered back in, earlier, even in the 60s and so on. But it was really not clear what they were doing. Histocompatibility means, tissue compatibility, but that's not, the reason we have them is not to reject each other's kidneys, right? So the real function is to present antigen to T cells, and that was discovered in the 70s, and then in the 80s it was clear that what they presented these MHC molecules to the T cells were little peptides. But it wasn't clear how they were made, okay? And then in the early 90s, just when I joined Per's lab, the sequencing of the MHC complex revealed that there are transporters in there and proteasome subunits. And so it's like a light bulb went off for the entire field where you realize, wow, that's how this works. The proteasome degrades the proteins, and the transporters then transports the proteins across membranes into the endoplasmic reticulum, and then they're loaded onto class 1. And so Per basically said, so you're going to work on that. And I said, that's great. There were just 4 Nature papers. None of them was from your lab. And he said, oh, we're better than these guys. So it was extremely competitive, but also a lot of fun. And a lot of my competitors from back then are still my friends. And it was really a very exciting time. So then 3 years into my postdoc, Per decided to join Johnson & Johnson and became basically industry. The whole lab worked for Johnson & Johnson then sort of overnight, which was good because it kind of gave me a lot of freedom. We had money from J&J, and I then focused on, picked up something that Per's lab had worked on back in the 80s on how viruses downregulate the MHC system. And in the 80s, that was the first one that was discovered was from adenovirus, E19. And there was 2 labs in Sweden. One was Per, the other one was Sune Quist. And I got interested in herpes viruses. There was a paper showing that there's a small protein in herpes simplex. And it turned out that protein blocks the peptide transporter. And meanwhile, we've discovered several, including pox viruses. Some of them have that. And so that was a very clever way for the virus to basically shut the door. The peptides can't be transported, they can't be presented, so it's a good way for the virus to hide from T cells. And then that led me to work on cytomegalovirus, a virus I still work on, because CMV has not just one, but 5 or 6 of these proteins, and they all do different things to the MHC system, and basically make the virus sort of invisible to T cells. And then I decided I wanted to go back to academia. So I didn't want to stay in big pharma because I also saw the writing on the wall. I cannot do basic research forever. And Jay Nelson, who I had known, started an institute in Portland called the Vaccine and Gene Therapy Institute, and had the idea that you would bring people together around a concept, of working together and, covering different aspects of research. And I really liked that idea because I had never written a grant. I didn't grow up in the system and, coming from industry. So being in an environment that had a lot of synergy was very attractive to me. And Portland is a nice place to live. I love the West Coast. And so we moved up there. And, ever since I've been at the Vaccine and Gene Therapy Institute at OHSU.
Good. So when did you move to OHSU?
In the year 2000.
Okay. Man, that was nearly 24 years ago.
It is. Time flies.
Crazy.
So today I want to talk about CMV and using it as a vector. But to start, give us enough CMV biology to understand what we need to know.
So CMV is a herpes virus. Now most people know herpes simplex, gives you blisters, and that's an alpha herpes virus. CMV is a beta herpes virus, and then you have the gamma herpes viruses that can cause cancer, like EBV and Epstein-Barr virus and Kaposi sarcoma virus. So CMV is a very large virus, the largest herpes virus that infects people, and most of us are infected and will never know it. Unlike the other herpes viruses that sort of hide in certain cell types, CMV is not only ubiquitous in the population, it can actually infect a lot of different cell types. And it never really shuts itself off. And what makes it interesting immunologically is that it, something's here, that it continuously stimulates the immune response. And so this became clear in the 90s, when people were studying human immunology. You look at various viruses that are ubiquitous, like EBV and so on. Everybody has that. And, you can detect immune responses. There was EBV, there was others like, measles or whatever. And then there was CMV. So this was like so much more easily to detect. And so it turns out that what CMV does, it stimulates an immune response, particularly a cellular immune response. So there's the antibodies, and then there's the T cells, and the T cell response to CMV is enormous and is always in circulation. So it's called an effector memory response, and these are not really long-lived T cells, it's long-lived memory. So they're constantly reminded. So the fact that CMV continuously reactivates at a very low level in your body stimulates these T cells. And then it was Louis Picker's idea, who also started around the same time at the Institute than I did, what if we could use this effector memory to control other pathogens or even cancer, right? And that's why we thought, so let's try out CMV as a vaccine vector.
All right, so this special kind of effector memory was the impetus for looking at CMV.
Was the initial idea. And the thing is, so all other vaccines basically elicit ultimately what's called central memory T cells. So what happens is you get vaccinated, we all got COVID vaccines and so on, and then over time, within a year or two, the T cells, it can take longer, but eventually they become less and less the T cells in your circulation. But they survive in your lymph nodes. Mm-hmm. When you then get infected, dendritic cells carry antigens into the lymph node, and then you get this explosive replication of these central memory T cells, and then you mount a recall response. With CMV, the idea was very different, that the T cells don't need to proliferate, they're already there, because they're not in the lymph node, they're in the periphery, and they are at a very high frequency. So a way to see it is like it's like an immune shield that is always there. And there's really no other viral vector that is able to do that. So it's quite unique to CMV.
These effector memory T cells, do they have physical characteristics like surface markers or something that distinguish them from other T cells?
Yeah, that's correct. So, to be precise, it's CD28 and CCR7 negative. What that means is that they don't have the receptors that are required to go into the lymph node. Okay. And so basically there is a number of markers. But the other thing that's interesting is, a lot of chronic viruses, for instance, hepatitis B virus, they make strong T cell responses, but then they become what's called exhausted. Mm-hmm. So they no longer respond. They become unresponsive, and that often happens with viruses that have a very high antigen load, which is like hepatitis B virus. Almost every liver cell is infected, and in many chronic viruses, the T cells ultimately then don't help anymore. Mm-hmm. This is not the case for CMV, so they're non-exhausted.
Well,
So I have a couple of immunology questions, but this might be me going into the weeds. Sorry. So you mentioned that you talked about the CMV inhibitors of antigen presentation, like the inhibitor of TAP.
Yep.
How do you think that ties into that very high T cell response? Because you would originally think that those two things would not go together.
Great question. I've got these questions ever since I published paper on class 1 downregulation. So how can you get these very strong responses on the one hand, when on the other hand, the virus is very, very good on hiding from the T cell it induces? So one way to think about it is that the induction of these T cells happens in a very different way than what the T cells then recognize. So it usually is not infected cells that induce those T cells, but dendritic cells that take up infected cells. And so this has very nicely been shown in the mouse CMV model by Ann Hill, where you can specifically in mice you can specifically knock out these cross-presenting dendritic cells, and then you can show that you lose some of the T cell responses to CMV. So one way is the induction of T cells, which is quite different from the hiding from the T cells that you induced. At the same time, we know that the T cells control CMV. And so one way to think about that is, when a virus enters the cell, it is not immediately able to shut off the alerting to the T cells. It's not immediately able to shut off the MHC system. It takes a while to make those MHC inhibitors. And that gives the T cells an opportunity to attack the infected cells. And, so in the case of CMV, therefore, a lot of the T cell response is focused on structural proteins and proteins that are made very early on, the immediate early proteins, and those are also very abundant. And CMV wants that. So one thing one has to realize is, you're not dealing with a hit and run virus. You're dealing with a virus that wants to be controlled by the immune system. And we had, a couple of years ago, we had this very interesting finding. So we knocked out one of those structural proteins. This was done in monkeys using rhesus CMV, and the virus grew better in the absence of this sort of immunodominant structural protein called PB65. So it's almost looked like the virus attracts the immune response and wants to attract it and wants to be controlled. So I think with herpes viruses and a lot of these chronic viruses, we have to think about a different paradigm. So this is not, a virus that, wants to evade everything. The virus wants to be controlled and it wants to get transmitted to others, but it doesn't want to cause any problems, so it's quite different.
The effector memory cells, do they show up in circumstances other than a CMV infection?
Yeah, so, you get them with, many other vaccines for a while. What makes CMV so unique is that they stay with you for life. So, CMV you often get with breast milk. Actually, breast milk is a great source of CMV. And you get chronically infected. And for the rest of your life, you will have these very strong T-cell responses. In fact, as you grow older, because the rest of your immune system kind of ages and gets weaker, the CMV responses take up a bigger and bigger percentage of your T-cell response.
I hadn't really thought about the effector memory cells being in the periphery and kind of that, the utility of that until you explained it the way you did. That was really helpful. Are there any particular anatomic locations in the periphery where those cells predominantly are? I could imagine if you were using this for a vaccine, targeting to different locations might be important.
Yeah, good question. So, you find them in the blood. Where they're particularly high is in the lungs, for instance. Liver also, spleen. So, there are some of the organs, they are higher than in others. But, we usually do either blood or lungs where we look at them. But, in the lungs, usually almost 10 times higher than what you have in the blood.
Now, there are circumstances where CMV just shows up with some pathogenesis, right?
Absolutely. And, the worst case scenario is if CMV gets into the fetus. And, just like with Zika virus, and if a virus gets into the fetus is completely, cannot defend itself against viral infection. It gets into the fetal brain. It can cause really big neurological damages. And so I'm actually working with a group here in New York, Sally Perma, where we use the monkey model to study this fetal transmission, because it's really difficult and there's no good solution at this point how to prevent that from happening. And for the families who have such kids, it's devastating.
And also in transplant patients.
And in transplant patients as well, even in late stage AIDS, so, which we don't see anymore these days, but in, the early days of the AIDS epidemic, they would get retinitis from CMV. In transplant patients, although in transplant patients, it's easier to treat. They have more option than in congenital infection. Once the virus is in the fetus, and this is often not detected very, very easily, so that's, to me, seems to be the bigger problem. Transplant, you have, various options. You have drugs, you have, adoptive T-cell therapy and so on. And so I think that's, it's still a problem, but it can be managed.
I assume that the issue with transplant patients is that they're immunosuppressed, right, for the purposes of the transplant.
That's correct. And one really clever way that I don't understand why it's not used more is to use what's called off-the-shelf T cells. So you can you can basically create a bank of T cells from people who are immune against not just CMV, but EBV, BK viruses, others that cause problems in the post-transplant setting. And because you're immunosuppressed, you will accept those T cells. And as long as one of the MHCs matches with the donor T cells, they can counteract the viral infection. And then as your immune system recovers, you get rid of those T cells.
Can you bank your own T cells before you get a transplant?
That's also a possibility, actually. Good idea. That will also solve a lot of problems. As long as you're CMV positive. Now, the problem is the worst outcome, of course, is if as a transplant recipient, you're negative, and then you get an organ from somebody who is CMV positive. So now not only don't you have immunity, now you're also getting the virus on top of it, and you're immunosuppressed.
So what fraction of the adult population is CMV positive? CMV negative must be pretty rare, right?
Not really. So we did a study in Portland in sort of preparation of clinical trials, and tested about 500 people, and pretty much exactly 50% of them were CMV negative and 50% of them were CMV positive. So it depends a lot on where you live, generally, but here in the US, I would say it's, 50 to maybe 70% of people are positive, but then in developing countries, it's 100%.
Mm-hmm.
So not having CMV is actually something very recent. It has to do with hygiene and so on. And so CMV has been with us, other than that, forever.
Too clean, man. Eat dirt.
Exactly.
So essentially, the virus is in a good balance with with these T cells,
Yeah,
which keeps it in check, doesn't let it get too high, not be
Correct.
pathogenic.
correct.
And if you alter that, then you have problems.
That's right. That's right.
Okay.
I mean, it does show that you need the T cells to protect, because when you don't have T cells, then CMV becomes a problem.
So these effector memory cells, the idea would be to harness them in a vector, right?
Correct. And what a vector is, for your audience in this case, is basically you use a virus to carry antigens from other pathogens. I mean, a good example, is the Johnson & Johnson vaccine, the AstraZeneca vaccine. They used adenovectors to express the spike protein of coronavirus, and some of us got these vaccines, and that's what you call a vector. Now, CMV was somewhat of a revolutionary idea, because most people thought it wouldn't work, because so many people are already positive. And from the adenovectors, we know, I mean, the reason why we use chimp adenovectors and rare adenovectors is because most of us have adenovirus immunity,
Right.
and then the vectors don't work.
Mm-hmm.
So I remember early on when we submitted grants and suggested it, people said, this will never work, because you have antibodies to CMV, you have T cells to CMV.
Right.
And well, it turned out it works really well. So we did most of our studies in monkeys, and all the monkeys have CMV. But you can go in with any dose. That's so surprising. With a replicating virus, even with 10 viruses or even less. I mean, at some point you cannot really accurately determine the titer anymore. And you will elicit an immune response to the inserted foreign antigen into your vector. And that's somewhat specific to T cells, because for antibodies it's a lot harder. You need a lot more, antigen and so on to get antibody responses. But T cell responses are very, very surprising. That you get those in CMV positive animals. And that's sort of when I started to work. So the whole idea of using CMV as a vector was Louis Picker's idea, and then initially worked with Jay Nelson, who had a long-term history working with CMV. And I got curious about this fact that he could so easily use it in CMV positive animals, because I had worked on these immune evasion genes. I said, I bet you if we knock those out, then you won't be able to use it in CMV positive animals. And that turned out to be the case. So we knock out those viral genes that downregulate MHC, basically enable the virus to hide from the T cells. And in CMV positive animals, no immune response. But in CMV negative animals, it still worked. So it really showed for the first time that this immune evasion actually really works in vivo. And the rest is history. Then I got more and more interested in this whole CMV as a vector, and Louis and I have a great collaboration ever since, and discovered very amazing immunology along the way. So this, we also discovered then when we knock out these immune evasion genes and put them into the CMV negative animals, the specificity of the T cells changed compared to the wild type virus. So that was the first indication that we can modulate what exactly the T cells recognize. And these are CD8 T cells, the typical killer T cells. And then there are the CD4 T cells, helper T cells. They're both induced to a very strong way. But the CD8s, we've now discovered ways how we can really modulate the specificity of those responses. So meaning that we can elicit T cell responses to different parts of the viral proteins. Okay.
Yeah. Okay. Sorry, my mind is a little bit blown now. So if you get rid of the immune inhibitors, specifically the antigen presentation inhibitors, you're obviously getting an immune response, or getting more of an immune response. And the pre-immune monkeys can now control better, and the vaccine doesn't work?
Yes. So
So you clear the vaccine?
exactly. So
Okay.
not you don't clear the vaccine never gets in.
Okay.
Okay. So if you basically knock out the genes that the virus has, or the proteins are ultimately, right, to hide from the T cells, you can no longer overcome the pre-existing T cell response. So that is one of the features that makes CMV so unique, and it's one of the reasons why it works in CMV positive animals, and hopefully also in CMV positive people.
But in CMV negative animals, it still works because they're not ready.
Exactly,
right?
exactly.
The virus gets the jump on it.
Exactly, and it established long-term infection in those, so it's not that it's going to be cleared.
And you change the epitopes?
And that was the other thing. So I mentioned earlier,
Yeah.
I mentioned earlier how, the wild type CMV, the T cells are elicited not by the infected cells, but by dendritic cells that take up the infected cells. So in the process called cross priming. Now we discovered that in these CMV negative animals, they got the virus without the immune evasion genes. We all of a sudden got new epitopes. In fact, those were the epitopes that all the other vectors would induce, even SIV itself. So we're putting semen immune deficiency virus antigen in there. And all of a sudden, these epitopes pop up that we've never seen before because the priming is now different. The priming now can also happen, the T cell priming from infected cells.
So it's sort of you're adding classical priming onto cross priming?
You got it.
Okay.
You got it. That's exactly what happens. But then that set us on a path that led to some very unexpected findings, and then we really wanted to know, so what are the epitopes that are being recognized, or what presents those epitopes? And we found out that all the others were not presented by MHC class 1 molecules, but by MHC class 2 and MHC-E. So now it gets complicated. So the MHC class 1 is, in human, it's called human leukocyte antigens, HLA-1. So these are the molecules that are expressed on every nucleated cell. That's the one that is associated with beta-2 microglobulin. It's very polymorphic, so a lot of us, each one of us has a different set of these HLA molecules or MHC-1 molecules. And that is what CD8 T cells, the killer cells, recognize. So in a way, this is a way for the cells to alert the T cells, like, look, I'm infected here. There's a peptide from the virus that's in the cell, and then the killer cell comes in and says, yeah, let's get rid of this. So that's every vaccine, everything that's out there, what we know is what usually those killer cells recognize. Now, in these CMV vaccinated animals, all of a sudden these killer cells didn't recognize class 1. They recognized a class 1 molecule that's usually not used to alert T cells, but to alert NK cells, completely different story. And MHC-II molecules, which are usually there to alert the other set of T cells, the CD4s, which are the helper T cells.
So these are CD8
T cells.
CD8 are restricted by class 2.
By class 2 and CD8 restricted, that's the word exactly, by MHC-E. And, this in the beginning was just sort of oddity. But, we did a lot of work trying to figure out how the CMV is doing that. But then what was really surprising, that particularly those MHC-E restricted T cells, that we need them to protect against SIV. And so that's the whole other part of this story. So as I said initially, the idea was for CMV used as a vector, you create this immune shield. And the idea that Lewis had was maybe that will help to protect against SIV, the monkey version of HIV. And his argument was that if you look at the infection curve with SIV, if you inoculate a monkey with very, very little, maybe 100 viruses, okay, within 7 days, the monkey will have 10 to the 6, 10 to the 7 viruses per milliliter of blood. So there's this explosive growth. So if you think about traditional vaccines, it takes 2 weeks for the T cells to have the recall response. By then, you already are, full with virus. So it's too late for the immune response to do something about SIV. And as you all know, for 40 years, we've tried to develop an HIV vaccine. There's nothing out there. Nothing worked. And, this might be one of the reasons. If you cannot completely block the virus from getting in, then it's very hard to clean up afterwards, okay? And because HIV integrates, it hides in certain immune privileged sites in the body and is very, very hard to get rid of.
So basically in 2 weeks you establish that reservoir and you can't do anything.
And then
right?
you cannot do anything, And interestingly, when you treat SIV, also HIV infected people very early on within the first week, you can prevent that from happening. So the idea was that this vaccine could work like an immediate mop-up, okay? So as soon as the virus, and also in the beginning, the HIV is also extremely variable. So there's, it also is not, you don't have as many different HIV sequences, so it's easier for the immune system to control that. So, and then Lewis, Picker, and Scott Hansen and others, they demonstrated this actually works. So in monkeys, that got vaccinated with the CMV vectors carrying SIV antigens, they got infection first with SIV, but then SIV was mopped out and was eliminated from those monkeys. What we didn't anticipate, okay, is that only worked when we used vectors that elicit these unconventional T cell responses, these MHC-E restricted T cells. That was unexpected, and that was a sheer coincidence because we picked a CMV vector that was based on a CMV strain that had been in culture since 1968 called 68-1. So a hippie strain, if you want. and had accumulated a couple of mutations. And that's the guy that induces these unconventional responses. If you would have used a wild type like vector that doesn't have those mutations, it wouldn't have worked. It would not have elicited those unconventional responses, and we would not have gotten.
So the CMV you and I have doesn't do this?
Doesn't do this, no.
Okay.
It doesn't do this. And also the CMV that monkeys have doesn't do this. Only the one that is deleted for certain gene regions. And it's 2 independent deletions that needed to happen. So talk about cosmic coincidence.
Yeah.
I mean, it's pretty incredible.
Okay, so only this deleted virus is presented on HLA-E.
Induces those HLA-E restricted T cells.
Or I guess mammo.
Yeah, mammo-E in monkeys, very good.
So is the antigen that's presented a CMV peptide, or is it something else?
Yeah, I mean, so basically everything, all the proteins that are expressed by these vectors will be, inducing T cells that are either MHC-E or MHC-II restricted. And the gene regions that are deleted, they all encode for chemokine-like proteins. We don't yet know how they inhibit those responses, but CMV actually doesn't want those responses from being induced, and has multiple inhibitors that prevent those responses from happening. And because unlike the responses that are classic, which we talked about, but the virus is very good at hiding from them. These responses, the virus cannot hide very well. So the virus doesn't really like it. Okay, so it has all these inhibitors, and just by chance we picked the strain that had those inhibitors, had lost those inhibitors.
So one clarification, with these various vectors, you vaccinate with them, you will you carry that vaccine virus indefinitely?
That's correct.
It becomes established.
Yeah.
Okay.
Yeah, very good point, Rich. I mean, so this would be the first vector that was ever developed that will stick around for the rest of your life.
And you need to use this lab strain. You can't use CMV from you.
No,
right now.
Yeah. So we designed now human CMV vectors, that have the similar deletions, and those are the ones that we're testing in humans.
That's the only change you make to the CMV is to put these deletions in.
Yeah, pretty much. Yeah.
Okay.
And I wanted to say one more thing, Rich. What was your question again?
My question was about the persistence of the vaccine vector.
Yeah, I just wanted to mention that there is only one vaccine out there that persists, right? And that's the varicella zoster vaccine, another herpes virus.
But that's not a vector. That's
a vaccine.
Just the vaccine itself.
So the vaccine, the VZV vaccine, persistent
Yeah,
neuronal,
not
It
the
establishes
not the
latency.
shingrix, but the
right.
chickenpox
Because that's
vaccine.
an attenuated
Yeah,
virus.
no,
Can you get shingles from that?
yes.
Yeah.
That virus can be pathogenic in like leukemic
Yeah.
people and so on. So that is, an attenuated VZV, but
Yeah.
that has still residual pathogenicity. Yes.
I understand they're considering using Shingrix even for kids now, right?
Why not? I mean, I don't know. I haven't seen any clinical trials yet, but I bet it'll work.
Yeah.
That makes sense to me. I'm kind of waiting for that.
Yeah, I've
Okay.
heard the same thing.
Yeah. So the idea
For the listeners, Shingrix is just a protein.
Yep.
Okay, as opposed to what we're talking about, a attenuated virus vector. So it wouldn't have any of the issues associated with a vector.
All right. So tell us about the SIV, CMV SIV in non-human primates. What you found with that?
Yeah. So SIV is in non-human primates is extreme. Certain strains of SIV are extremely aggressive. You inoculate them at a very low dose, and then all the monkeys will come down with AIDS at some point. Sometimes you have spontaneous elite controllers, but generally, monkeys will get AIDS, and they will get AIDS very rapidly.
Mm-hmm.
So it's actually more aggressive than HIV. Okay, this is this SIV MAC239 strain, and it's been really hard to vaccinate against that. So a lot of the vaccines that have been tested in people actually don't work against SIV MAC239, because as soon as, unless you block the entry and you really block the virus from getting into the monkeys in the first place, once they're there, it's practically impossible to clean it up. And our work now suggests that, because they have the wrong T cells. You need, for some reason, and we don't know why, those MHC-E restricted T cells are the ones that control the virus. The others didn't work, even in our hands, right?
Sorry to interrupt. Are the MHC-E T cells or the predominantly MHC-E T cells related to the fact that they're effector memories, or the effector memories just based on the sort of kinetics of the virus?
Yeah, I think it's both. So the initial idea is certainly correct, that you need those effector memories to create this immune shield. But what we didn't anticipate is that they need to recognize SIV-infected cells via MHC-E and not via classical class 1 molecules. So it's the combination of the two.
So in those animals with CMV or rhesus CMV with SIV proteins, what was the result on challenge?
So, so they get challenged, and what we had was approximately 50 to 60% of the animals control SIV, and they control it so well that if you go back 1 year later and try to find virus, it's gone.
No virus. Okay.
No virus, even if you transfer, millions of bone marrow cells into a naive animal, where when you do this with SIV-infected animals that are on the antiretroviral therapy or these elite controllers, they always have virus. So you transfer it to a naive animal, it will get SIV infection. In these animals, no, it's all gone. So by any means, we can no longer detect it. The other half of the animals that got challenged, they show no signs of protection. And despite the fact that they have also the MHC responses, so immunologically, they're not different. And we have some indication what could be the reason there. So Louis Picker and Michael Gale teamed up and basically just cast a very wide net and just looked at all the transcripts that are expressed in the blood of the protected versus unprotected animals before they were challenged. So was there anything that could predict, which ones would be protected and which ones weren't? And there was a signature in the blood that looked like interleukin 15 signaling. And they actually showed by injecting monkeys with IL-15 that, a lot of those transcripts that were upregulated in the protected animals were in this pathway. And so there is something else going on in these animals that has nothing to do with the vaccine, but has to do with their innate setup, their innate immune setup that makes them being protected versus non-protected. We don't yet know exactly what it means and how we can modulated to get to 100% protection. But it's also important to remember, because this SIV is so aggressive, you cannot assume that the same will happen in people, because it's a different virus. It's human CMV, it's HIV, sure. So we don't know, if we are able to elicit those responses in people, how it will translate to protection against HIV.
With the IL-15 signature that you mentioned, is that something that is a sort of intrinsic genetic difference in those animals, or were their immune systems just having a bad day the day they got vaccinated?
I tend to think the latter. Okay, so because it turns out it's the ability to respond it's the difference between the set point before vaccination and then after vaccination, what happens. And what's interesting is that this IL-15 response stays up, the whole time. Whereas if you treat animals with IL-15, it's induced, but then it's also immediately shut off. So it's a very transient response. So for some reason, CMV is able to induce and maintain these IL-15 responses in these animals for the entire time.
I presume you want to do this in people. With HIV. So the non-human primate work was done a while ago. So what's going on with the humans?
So we're still working on non-human primates, and we want to know, what's up with these MHC restricted T cells. How do they control SIV? Because why is that? What is so unique about them? On the one hand, and on the other hand, how does CMV induce those? And is this something that potentially we could translate into another vector system? And at the same time, we started making human CMV vectors, and we started this quite a while ago. And as I mentioned, introduced the same deletions and so on. And this has now all been transferred to Vir Biotechnology, a company that we founded a number of years ago, or co-founded. And the entire clinical trial is now run by Vir, is sponsored by the Gates Foundation and by the NIH, and is also done by the HIV Trials Network, the HVTN, which they are both here and in South Africa. And I'm happy to report that the first patients have been dosed, and we'll keep the fingers crossed that we see something, and if we see something that is HLA-E restricted. So, we're waiting for the results, and it's a very exciting time for all of us. I mean,
So this is this is a phase 1 trial then?
It's a phase 1 trial in HIV negative, and they are CMV positive. And so we basically go into people who already are chronically infected with CMV, and also to test whether the same is true in people, that you can overcome pre-existing immunity with CMV vector.
So it's a small trial, I guess, if it's a phase 1, right?
It's a small trial, and we should know the answer relatively soon.
But you don't think any of them will be infected with HIV in the meanwhile, right?
No.
It's just safety trial.
Yeah.
Right?
This is just safety and immunogenicity, just to see whether we get the HIV-specific responses induced by the CMV vectors.
I'm thinking, however, I have to say this, that the subjects in that trial now, from everything you've said, will carry this CMV vector for the rest of their lives.
That's correct.
And whether that's an issue, we don't know yet, right?
Yeah, well, I mean, you have to think about that. You don't give them something that they don't already have, so they're already infected with CMV.
They've already got CMV, right?
And now you put the CMV in that lacks a bunch of genes that has no chance of competing with the endogenous vector, endogenous virus.
Yeah. Okay.
How far into this trial are we?
We just dosed we meaning the HVTN Trials Network. They just dosed the first patients.
So let me go back again, because I'm to the perhaps the non-human primate model. If you take a non-human primate that has CMV and administer one of these vectors, what fraction of the CMV that animal carries down the road is vector, and what fraction is the CMV they started with?
I have no idea. You know,
Okay.
what we do know is different from people. Monkeys continuously shed CMV, so you can always detect it in the urine, and so you can always see that if they're CMV positive, you know, you can detect virus in the urine and in the saliva of these animals. And we do know that our vectors show up at some point. And we can detect them, and we can detect them for a long time. Quantitatively, it looks like there is less vector than endogenous virus because, as I told you, you know, it has a bunch of deletions. You know, so it is not as good in probably in replicating. It also cannot infect all the cells that the wild type virus has. So based on that, I would say, you know, that the majority of virus you have in your body or a monkey has in their body at this point is still the wild type and not our vector. We also know from other studies, you know, where we used, a lot of my work over the last, 10 years or so was also geared towards safety, you know, so can we make these vectors, how safe can we make them, you know, and what's interesting is you can really uncouple any kind of pathogenicity from immunogenicity. So you can make vectors that basically do not spread, okay, that are stuck in the first cell they infect. And we need, about 10 to the 5, 10 to the 6 of these to elicit an immune response. And they won't elicit antibodies, even to CMV, even in CMV naive animals, but they make very nice T cell responses. And in at least one case, which we haven't published yet, we also can protect against SIV with these very safe vectors. But what that means, if you think about it, so you go in with 100,000 or 1 million viruses or vectors, and you inject it on subcutaneously. So there's only a couple of thousand cells that ultimately will be infected, and that can elicit these very strong and maintain these very strong immune responses. So we don't know which cells they are. We have some ideas, and we're following up on this, but it's a remarkable feature if you think about it. Even with such highly attenuated, non-spreading CMV, that infects a couple of thousand cells, you get these, several % of your T cell responses will be CMV specific and will remain like that.
Yeah, that's my big question. I guess you don't know if it's going to stay like that, because I have, my first reaction is, well, you would clear that eventually.
We've never seen clearance. Now, the point is that, effector memory indicates that sort of it's not long-term memory cells. They are cells that have been recently reminded. Okay, so at the very least, we need long-term antigen presentation somewhere. Okay, and so based on the T cell phenotype, we have never found anything we do to these viruses. I mean, the deletion screens and what have you, which would eliminate the ability to maintain those effector memories, which in turn means that the virus must stick around somewhere. And in this case, when we have these non-spreading viruses, it can't go very far. And so it seems that CMV is just very good at what it's doing, and it just needs a few cells to be infected, and that keeps the immune system just stimulated well enough to keep going.
So are there limits to this? You've mentioned, you can use this in people who are already CMV positive.
Yeah.
But could you vaccinate against one pathogen using a CMV vectored vaccine and then use the same CMV vector to vaccinate against another pathogen?
Yeah, and that's exactly what we're doing, because monkeys, of course, are very expensive. And so, we use them repeatedly in studies, and we can always add additional T cell responses by changing the antigens that we put into the CMV vectors. And it's a good question, Brian, and because there is competition for space in the lymph node. So for other vaccines, so there is a competition, because you get these central memories that compete for space in the lymph node, but that's not what the CMV-induced T cells are. So in the periphery, you can always add additional T cell responses. Now, the frequency in terms of percent of the total response goes down. Let's say in the beginning you have 10% are SIV gag responses, then you add some TB antigens or something, then those are 5% of the total response, but the absolute numbers don't change. So there is a lot of space in the peripheral response, a lot of space in the liver for T cells. Ultimately these are still relatively small numbers compared to liver cells or other cell types.
So would there be use of these vectors? So you're talking about, for SIV at least, rapid clearance before latent reservoir is established. So would this be useful for SARS-CoV-2, where, say, a variant is evading antibody responses, but maybe T cell responses would keep it in check within a week or two? Is that possible?
It's a good question. So the T cells, of course, only work after you get infected, because they need infected cells, and then you have this antigen presentation. So you won't prevent infection. And in the case of SARS, you could imagine that this would give you a long-term immune shield, but it would be T cells. Okay, and we know, you guys are the experts better than I am. We know that antibodies are also very important. And so I think the T cell protection by itself might not be sufficient, but it would be worth testing.
It is for SIV, right?
For SIV, but it's also this peculiarity that, in the case of SIV, when the immune system kicks in, it's too late because the virus has already integrated and so on. And, whereas in SARS, by the time you're vaccinated, your immune system kicks in, the immune system will be able to wipe out the virus. So
but it's worth looking at for SARS-CoV-2.
It would be worth looking at. But generally, I think, you have to think about this as a vaccine platform for situations where having this long-term immune shield, would be great. Think about cancer, for instance, So in many cancers, the primary tumor is removed. And then for the rest of your life, you walk around worrying about having, a little piece of that tumor metastasis in your body. So if you had a CMV vector that is specifically tuned to recognize those cancer cells, and then basically mop up all those metastases in your body, because that's what those T cells do. They are all over. You wouldn't have to worry. And this is particularly true for viral cancers, where there are a lot. There's papillomavirus, and Epstein-Barr virus, and so on and so forth, where you could create this sort of lifelong immune shield. And I think for those applications, that's where the CMV vectors, I think, have the best application.
What other infectious diseases have you tried with these?
So we tried TB. We had very good success with tuberculosis in monkeys. And again, it's pure T cell vaccine. And then I did a study with malaria also, where we still got infection, but we saw a delay of infection. And the way to interpret that is, the T cells cannot do anything against malaria once it's on red blood cells, because red blood cells don't have MHC molecules. So they can only interfere with malaria at the liver stage, which is where you want to interfere. But the main problem in the malaria field is, as I told you, malaria was my first love, so I still follow the field somehow. The main issue in the malaria field is that we know very little what actually happens in livers, in infected livers, because there's no in vitro model for it. And so nobody can tell me, what is actually presented on infected liver cells. So we recently started a collaboration with a woman from Brazil, Caroline Junqueira, who just accepted a position in Bellinzona, Switzerland. What she has done is she went into Plasmodium vivax infected people. So vivax, unlike falciparum, also infects reticulocytes. Reticulocytes are the precursors of red blood cells, and they still have MHC molecules. So she was able to isolate the peptides from those MHC molecules, and for the first time really sequence what's being presented. And so now we're collaborating,
Nice.
and interestingly, some of them seem to be presented by none other than MHC-E. So we're now really excited. And so together with Brandon Wilder at our institute, we're now testing, whether we can design a vaccine that would specifically target what is actually being presented. Because that's been my question to the field for the last 10 years or so, and nobody can tell me. So I'm very excited about that.
These vectors are very exciting.
They are, and we just, fingers crossed for the human clinical trial. I think this could be a real, new platform that's unlike anything that is out there. And the sky's the limit once we basically broke the sound barrier of human clinical trials.
Yeah. So this first phase 1 in humans is really important.
It's really important, yeah.
Now, great.
And when do we get results from that?
I can't speak for Vir, okay? So, but the trial's ongoing. It takes a couple of months. I mean, the enrollment is not that easy because people have to be CMV positive, non-childbearing age, blah blah blah. So it's quite restrictive of the people who can partake. But I think it's a very good network that's doing that, and so we should get results sometime next year.
So I'm sorry, I need to go back and remind me. This was an HIV antigen expressing vector or just
Correct.
vector alone?
Correct.
HIV
it expresses HIV antigen, and the design of the HIV antigen is quite interesting also. So this is scientists at Los Alamos. I call them rocket scientists. Okay, that designed an antigen. So one of the problems with HIV, of course, is that it's extremely variable. It's a quasi species. So basically, what do you pick? What's your antigen? It's like there's clade A, B, C, and there's different regional strains. So Betty Corber at Los Alamos years ago started to work on something that she called mosaic antigens in the beginning, and then they improved together with her husband Jim Tyler. They improved this method, and then now they call it epicensus. And so what that is if you think about a consensus sequence, so consensus sequence is all the sequences out there lined up, and then you say, so what's the most frequent amino acid at position 1, what's the most frequent position 2, and so on. So what they did is they did that for T-cell epitopes. So as I said earlier, these are peptides. Usually for class 1 molecules, they are 9-mers. So what's the most frequent, epitope, potential epitope from position 1 to 9? Now, okay, if you do 1 to 9, 10 to whatever, 18, then it's easy. Because then it's the same algorithm as the consensus. Where it gets complicated, as you can imagine, is when you do 1 to 9, 2 to 10, 3 to 11. So, and don't ask me how it works, okay? So, but they developed an algorithm that can optimize this. And that's the basis for the insert that is in this HCMV construct.
Got it.
So would this insert actually ever be able to make a GAG protein,
Yes,
or is it just sort of Franken protein?
These are all epitopes from GAG?
Yeah, well, it's GAG, NEF, and a piece of the polymerase, okay? And so, the interesting thing is that, these are not oversized proteins. So these are basically proteins that could exist. Okay, so they just have the optimized sequence, but they're not, longer or very different. So they are basically functional proteins, just using the most frequent sequences in all positions.
And is there any evidence that they are going to elicit an appropriate response, or is this the test?
This is to some extent the test, but we've done studies in monkeys and they do work. So you can get immune responses in monkeys using the human vectors, but human vectors don't really replicate in monkeys. So they are not the same as the ones we get with the CMV vectors. But using those, we can actually show that we get responses to to these inserts.
So
something occurs to me, because all these vectors are still susceptible to the anti-herpes drugs. Is that correct?
Yeah, that's correct.
I would imagine that's part of your sort of protocol, is that if something goes wrong, you've got a backup.
In fact, the vector backbone we used was originally drug resistant. That's how it was isolated back in the early days of the AIDS epidemic. And we re-engineered the drug sensitivity into them. However, by treating with drugs, you do not eliminate the virus, okay?
Right.
And you do not eliminate the immune response, but you will be able to control the virus.
Right.
So remind me again how you identified these deleted CMV that have this unusual property.
Yeah, so basically this hippie strain,
68-1,
okay, had these deletions, and then we repaired those deletions. So compared to a wild-type CMV, it was very obvious that, there's something happened. There was a whole gene region that was inverted, and gene regions were deleted, and so we basically, and in fact what happened was somebody, Jeff Cohen's lab, in fact, sequenced the original isolate. Okay, there was still a sample available somewhere. So we knew exactly how this looked in the beginning, before the virus, over many passages in vitro kicked out these gene regions. And so we re-engineered that back in. And then we saw, okay, now we lost those responses. So we knew that the wild type wouldn't do it. And then we put those genes back in, and I should give a shout out to Daniel Maluli, who's doing all this work, and he did that, and so re-engineered those genes. And actually, some of the work was done even in Tom Schenk's lab early on. He had a student or a postdoc working on rhesus CMV. We used those vectors, and that had these gene regions repaired, and we completely lost these unconventional responses. We revert back to the conventional MHC-I responses, and that's what we see with a wild type.
So this was in a rhesus CMV.
This was a rhesus CMV, and just last year we published a paper where we recapitulated the whole system in a different monkey strain, in cynomolgus monkeys, or different monkey species. Also a little bit of an anecdote there. So these are Mauritian cynomolgus monkeys. Okay, the island of Mauritius in the Indian Ocean didn't have any monkeys until some Portuguese sailors brought in a small group. Apparently, it was only one female, and they became a pest. So the island actually tries to get rid of them. But over hundreds of years, it's basically a naturally inbred population that exists on the island of Mauritius, and these are not rhesus monkeys; they're cynomolgus monkeys. And different from rhesus, they have a much simpler MHC, much more similar to humans. And Jonas Sasha at our institute then had the idea, let's see if this works in these monkeys. We said, okay, let's just use our rhesus CMV and we vaccinate them, and then we challenged them with SIV. Well, the first surprise was the rhesus CMV first didn't infect, so we had to repair some genes to infect. Then it didn't induce those unconventional responses. And so then Daniel went back in and isolated cynomolgus CMV from the urine of these animals, converted into a vector, deleted those gene regions that we knew would be inhibitory, put in the SIV antigens, and now we're getting the responses, but only in cynomolgus monkeys, not when we go into rhesus monkeys, okay? And those responses, Jonah then showed that they are protected against SIV MAC239 challenge. So that was a very nice study.
And these deletions arose in passage in cells and culture.
Yeah. I mean, these big DNA viruses, they often, over years in culture, they kick out gene regions because you're picking the ones that grow the best. And there's one gene region that's called the pentameric complex, which is needed for infecting any cell but fibroblasts. But because you grow it in fibroblasts, that's often kicked out because you get more virus in the supernatant. So we're selecting for those deletions. Unwittingly, in this case, we selected for just the right deletions.
Amazing.
What kind of cell? It was rhesus CMV, and what kind of cells?
Rhesus fibroblasts, so they're grown in rhesus fibroblasts, And then it was Peter Barry at UC Davis and William Chang who cloned this 68-1 strain and gave it to us, and that's what we used in the beginning to make these
This
vectors.
is why you just need to let scientists follow their curiosity.
Yeah.
Right?
Well, another lesson is sometimes you just need dumb luck, right?
That too. But, following a path is important rather than trying to cure something. So the monkeys that went to Mauritius, they were originally what kind?
They were cynomolgus monkeys. You know, so the
Okay.
cynos are a little smaller. You know, you know, most of the monkeys we have at our primate center, you know, they're bred there, and they were brought in from India in a small breeding colony in the early 70s, and so they're Indian origin rhesus monkeys. And you know, cynomolgus monkeys are closely related, as are their CMVs. So it was quite surprising that this wouldn't work across species. And we're currently actually doing some cool experiments trying to figure out, you know, what exactly is responsible, you know, which genes for this species restriction.
I asked because we have an email that we haven't read yet from Jens Kuhn. You know him?
Yeah.
Who is talking about people calling them cynomolgus macaques is not right.
Oh, okay.
Because they're not macaques, they're cynomolgus monkeys, which is what you said.
Okay.
But they're in genus Macaca.
You got to talk to Jens.
Yeah, that's true.
Jens is listening.
Yeah,
he is.
We'll
He always
get more
listens.
mail.
He listens. We have to read his email one of these days.
So,
All right.
Yeah.
All right, that's great. I just think that's a wonderful story. And
I now have quite a few penguins on my iceberg. I'm going to have to think about this for a long time.
Yeah.
This is a lot of stuff.
Yeah.
I honestly, if I didn't have things going on this weekend, I think I would just read papers about this.
Yeah.
I am so ready to geek out about this right now.
So we just published a review article earlier this year, Louis and I and some others that covers this. Actually, don't remember was it
So
current immunology or something. Yeah.
You know, tell me if this makes any sense. My impression just overall is that CMV almost creates a special immunologic environment.
That's correct. There's
that you're taking advantage of with these vaccines.
Yeah, that's correct. There's a great study by Mark Davis, who did a twin study. Okay, so looking at twins who were genetically identical, but one was infected with CMV, the other one was not. Their immune system is totally different.
Wow.
Okay, so you can basically tell if somebody has CMV by just looking at their T cells and their NK cells. Somebody once said that CMV turns NK cells into T cells and T cells into NK cells. What they mean is you get this expansion of NK cells also. So the natural killer cells, which are non-specific, expand very strongly in CMV positive people, and they have certain surface receptors. It's probably a combination again of actually HLA-E being upregulated by CMV, but also the antibody response that CMV triggers. And so you can tell just by looking at the NK cell profile if somebody has CMV. And the same for the T cells. A large part of the T cells are these effector memory T cells. And now we have these very strange, T cells that recognize these non-polymorphic MHC molecule, at least in our vectors. And so this is absolutely correct. And so another way to look at it is, CMV has been with us even before there were humans and has co-evolved with us, but our immune system also co-evolved with CMV. And this is just remarkable. There are certain immune stimulators, like certain MYC proteins, for instance, that only exist in certain human populations, but CMV has already developed counter mechanisms against those. So this is not a static situation. This is still ongoing. Okay, how CMV teaches the immune system, and then the immune system teaches CMV what to do. But you're absolutely right, Rich.
So is it fair to say then as well? I'm trying to summarize all this in my head. It's fair to say then as well that harnessing that CMV specific immune environment may be particularly advantageous for managing certain types of infections like HIV, like malaria, like TB.
Yeah, I think so. And, particularly chronic or recurrent infections where you want to have this long-term immune shield, I think will benefit from this.
Man, okay.
Good stuff.
I got chills. I now officially have chills.
Nice.
Very good. All right, that's TWiV Chill 1057. 1057. You can find the show notes at microbe.tv. twitter.com/TWiV. You can send questions and comments to twiv@microbe.tv. And as I said before, if you like what we do, consider supporting us. Microbe.tv/contribute. Our guest today from Oregon Health Sciences University, Klaus Früh. Thank you so much.
Thank you, and I really have to thank you for what you're doing. I mean, this has
pleasure.
been, such an important podcast. Many people listen to it, and most of us don't do a good job at going out and spreading the word. So thank you all for doing that.
Yeah, you're welcome. We have fun. We're having a lot of fun doing it.
We do have fun. Yep.
Rich Condit is an emeritus professor, University of Florida, Gainesville. Thank you, Rich. Oh, sorry, currently in Austin, Texas. Thank you, Rich. Can't forget
Sure
that.
enough. Always a good time, and great to talk to you, Klaus. Good to see you.
Good to see you.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It's actually BioProf Barker on Blue Sky now too.
Ooh.
Blue Sky.
Blue Sky seems kind of like what Twitter used to be. I kind of like it.
Twitter doesn't exist anymore, right?
Well,
Now it's X, but Blue,
you know.
Blue Sky kind of seems like the old Twitter.
Yeah, we'll do Blue Sky. Okay.
I heard X referred to recently as the social network formerly called Twitter.
Yeah, that's the one. In any case, thank you so much, Klaus. This was clearly very much the best part of my week, and I learned so much. Thank you.
Thank you, Brianne.
Finally I get 175,000 followers on Twitter, and now it's no good anymore. All right, we'll have to work on Blue Sky.
Tell Rich's picture just got posted.
Oh, your picture just got posted, Rich. We're on Blue Sky or on
No, Instagram.
Instagram. Check out Instagram. Your
I will.
picture
I'll
just
do that.
went
Doing this.
up. All right.
My superhero picture?
Your superhero picture.
Okay, good.
Yeah.
I'll have a look.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.